Assalamu alaikum. I am Shihad Al Shariar from Southeast University. Today I will discuss about our research topic on effect of XRCC1 gene polymorphism on response and toxicities of chemotherapy in Bangladeshi breast cancer patients. So here are the authors and here are the key authors. Without them we couldn't complete this research. Moving to the next slide, the background of this research. Breast cancer remains a major health concern in Bangladesh, with chemotherapy being a cornerstone of this treatment. The XRCC1 gene or X-ray cross-complementing gene, group 1 gene plays a crucial role in DNA repair, and its polymorphism may influence chemotherapy response and toxicities. What are the objectives of this study? This study aims to evaluate the impact of XRCC1 gene polymorphism on chemotherapy response and treatment-induced toxicities in Bangladeshi breast cancer patients, providing insights into personalized treatment strategies. Moving to the next slide, breast cancer. What is cancer? Cancer is an unwanted proliferation of cells, and breast cancer is one of the most prevalent malignancies worldwide, and it is a leading cause of cancer-related mortality among women. Genetic mutations that compromise DNA repair pathways are critical contributors to breast cancer carcinogenesis. Among the DNA repair genes, the X-ray repair cross-complementing group 1 or XRCC1 gene plays an important role in the base excision repair or BER pathway, which is responsible for single-strand DNA breaks resulting from oxidative stress, alkylation, or other endogenous or exogenous DNA damaging agents. Moving into the next slide, here we can see graphical data, and here we can see on the right side that breast cancer from the other cancer, the mortality and incidence rate are too much high. And if we see the here pie chart, from other diseases, breast cancer is here about 16%, which is greater than other diseases. So we can see that the breast cancer is very critical in our worldwide. Treatment individualization. The ultimate goal of these genetic and pharmacogenetics studies is to enable the selection of the treatment that is most likely to provide benefit and minimal toxicity to the patients. So we are working on breast cancer from 2009, but this study is on XRCC1 gene is about 2 years, and we have collected 700 plus samples and we reserved in our laboratory, but we work only on 400 samples, and the number of patients receiving neoadjuvant chemotherapy is about 182, and the number of patients receiving adjuvant chemotherapy is 218, and the drugs use cyclophosphamide, epirubicin 5, and fluorouracil, what is called SAFE, and CEF, and cyclophosphamide, doxorubicin, 5-fluorouracil, and CAF. These are the combinations that are used to the patients as chemotherapy. Response assessment. We assess the response of the patients by response evaluation criteria in solid tumors or resist. By this method, we evaluate assess the responses, where complete response, partial response, and progressive diseases and stable diseases are measured in our study. And we XRCC1 gene or X-ray repair cross-complementing protein is an essential DNA repair protein in human. Encoded by the XRCC1 gene, it plays a crucial role in BER pathway, facilitating the repair of single-strand breaks. XRCC1 gene plays a crucial role in metabolism and DNA repair response to cyclophosphamide, and alkylating agent widely used in chemotherapy. Cyclophosphamide induce DNA crosslinks and single-strand breaks, which require efficient efficient repair mechanism to maintain genomic integrity. So here we can see genetic polymorphisms. So we work on SNP or single nucleotide polymorphism. Here we can see a graphical image where what is SNP, and we use to genotype the genotypic substance. We used the method is, firstly we collected blood from the volunteers, then we isolate the DNA from blood samples, then we do PCR to amplify the DNA, and then after that we use the PBU enzyme to as a restriction enzyme to digest the PCR products, and then we use 2% agarose gel to show the to get the results. So here are the PCR parameters. Here we can see that we use master mix for primer, reverse primer, nucleus-free water, and target DNA. These are total 20 microliter of a single PCR product. And here are the PCR conditions. And after primer design, we do primer design. Our chairperson, Siddiqul sir, helped us in this. And while doing the PCR, while doing the enzyme digestion to the PCR product, we incubated the product about 24 hours in incubator in 37 degrees Celsius, and we use the polymerase chain reaction RELF of XRCC1 gene. Here we can see a targeted SNPN, and green is indicates forward primer and yellow is the reverse primer. And when N is changed to C, then it shows a fragment about 485 base pair, which indicates normal homozygote. And when N shows C, but it maybe shows C into C or T, then it shows 3 fragments about 485, 396, and 889 base pair, and it is mutant homozygote. And when only T, it shows only 1 fragment about 89 base pair, and it is mutant homozygote. So here are the PCR results of the digestion product. And here we can show data how we work on this addition of gel samples and about the gel electrophoresis. And here are our image about the PCR product, where we can we found our PCR product on 485 base pair, and here is the digestion image of the enzyme digestion image. And here is the demographic distribution of the patients. Here we selected different data about age, about less than 45, 45 to 55, and greater than 55. And we also evaluate their education about the data. And here all are the demographic distributions of the patient. We study their data, and we compare here about, and here are a table. And after here the statistics we study from our research, we get CC genotype about 39%, and which case number is 156, CT about 40.5%. Which case number is 162, and TT about 20.5%, which case number is 82. And here is the correlation of XRCC1 gene with clinical pathological characteristics of the patients. Here we also measure the data in age, menstrual status, and TNM stage, lymph node status, different, but we didn't get any significance from them. And also we see the histological and tumor grade, and we also couldn't find any significance from them. And here all the correlation of XRCC gene hormone receptor status, just as estrogen receptor it is negative or positive, or progesterone receptor it is negative or positive, or HER2 new status it is positive or negative. All our data are evaluated, but no significance. We couldn't find any significance. Here we can see the response variation of patient with XRCC1 gene 1 polymorphism. So in the neoadjuvant, about patient was 182, but the total responders are about 106, and non-responders are 76. So the responders about CC genotype was 36 and 34, and CT was 45, and non-responders are 30, and TT was 25, and non-responders are only 12. But here we also couldn't find any significance. And if we here we see the anemia in both adjuvant and neoadjuvant chemotherapy. So here we the slide starts of toxicity evaluation. So in toxicity evaluation, we see here some data we get from the patients, and here anemia grade 1 and 2, that is shows as less than 2, and grade 3 and grade 4. So after evaluation, we can see that here we find significance of TT genotype and CT plus TT genotype. So we can see that we can say that anemia can be happen if XRCC1 gene mutations happens. And after that, neutropenia, here we can see same method at grade 1 and 2, 3 and 4. And after the data evaluation, we can also find that neutropenia can also be happen. Here TT and CT plus TT genotype is responsible. And after the leukopenia, about the leukopenia, we can see that CT, TT, and both CT and TT both are significant here. So these are responsible for as a it can shows toxicity. And as thrombocytopenia, here we couldn't find any significance. And about the gastrointestinal toxicity, we evaluate the data, and we only find about when the SNP is changed to TT, and about the combination of CT plus TT, it shows significance. So it is responsible for the toxicity. So this study and this study of the findings of XRCC gene is related with chemotherapy-induced toxicity of anemia, neutropenia, leukopenia, and gastrointestinal toxicity. Response variation is not assisted. The polymorphism of this gene, no other association with clinico-pathological characters of demographic distribution was found in the study. So the recommendations are that if we do gene polymorphism studies on the vast of patients and find that, and do whole genome sequencing, we can find that what chemotherapy is responsible to show toxicity. So we can do personalized medicine for the patients. Thank you so much.